you can go ahead.
Thank you. Thank you, first of all, for the interesting presentations. I had a question in mind that's probably mostly related to Professor Sedlak, but it can be answered by anybody in the panel, which is how could we tackle the arising issue of retentate produced from reverse osmosis processes, especially in places where the retentate is just pumped back into smaller water bodies like lakes and gulfs and rivers. Thank you.
Great. Well, thank you for that question. That actually is something that keeps my group up at night working on, and Anthony, who introduced me, is studying that for his dissertation. So those of you who, I didn't really talk about this in the discussion, but reverse osmosis is a wonderful technology. It removes just about everything from water. It makes clean water, but it produces a waste material called reverse osmosis retentate or concentrate. When reverse osmosis is used for seawater, it's just concentrated seawater. It's like double the salinity, and people usually just mix it back into the ocean. Sometimes that's a problem, usually it's not. But when we use reverse osmosis for treated sewage or industrial waste, we essentially concentrate all those wastes into a smaller volume. And so we're finding that this is turning out to be a limitation in water recycling, and we need to develop technologies for treating this concentrated mixture of organics and inorganics. And Anthony, who introduced me, is studying a nature-based system. So we have a subsurface wetland in San Leandro, where we're studying the ability of microbes to break down chemicals as they flow through the subsurface. But there are other researchers using ozone and electrochemistry and the classic oxidation technologies. And the advantage is that you're doing this in a smaller volume, and so you can have a greater intensification of the treatment processes. Finally, the last thing that's possible is so-called zero liquid discharge. And in a project with the National Alliance for Water Innovation, or NAWI, at the Department of Energy, we're looking at ways to actually remove all of the water, and then we'd be left with a solid material that either could be disposed of in a landfill or subjected to additional treatment for a lot less money.
So before we move on to the next question, Anthony, do you want to add anything to that? It's good? Okay. All right, we have a question over here.
Hey, David. I'm Yao. I'm a postdoc here. I did my PhD in electrochemistry at Cornell. So my question goes to the electrochemical peroxide production. So if you want to make into an electrolyzer, you need a membrane to separate the cathode from anode. Can you comment on the stability of the membrane in response to the peroxide and also hydroxyl radical?
Yeah, so, and again, I encourage you to talk to Yanghua Duan, who's over there in the audience afterwards, because this is his dissertation topic. Okay. We've been using a 3-electrode system where we separate the cathodic production of hydrogen peroxide from the activation on the stainless steel. And so we avoid some of the damage that occurs when hydroxyl radical reacts with the membrane. Mm-hmm. And then we have a third system where we have an anode, and that anode completes the circuit. But this is one of the things that we're spending a lot of our time on now, making sure that the oxidants that we produce are not damaging our membranes and our electrodes. And that's also the other, of course, as you said, the other weak point of these systems quite often is the membrane used to separate the cells. And there's a lot of, in the engineering of an electrochemical treatment system for low conductivity water, getting the inter-electrode distances quite small, and that's why it's only really appropriate for small-scale systems. So happy to talk more with you, but Yanghua knows a lot more about it than I do, probably.
Thanks.
I have a question for Bengt. Could you comment about the lifetime of this stretched DNA, and then as you stretch it, you're making spaces within the strand? Is there anything that, do these spaces have a role in this process? I'm interested in the lifetime. And then do you see any bending? Is it, what kind of perturbations do you see? And what I would also want to ask more about, what is the role of this stretched DNA? Does it have a function?
Well, the stretched DNA, as we see it in single molecule experiments, that's an artificial structure. And of course, you could think of the possibilities that it's used. Nature is discovering everything, so you could imagine that fluctuations in nucleic acid, including RNA, are transmitted along the nucleic acid and is focused into large amplitude stretching effects. And the confining effects is due to the backbone. So if you choose between a homogeneously stretched DNA, we know that it's 50% stretching, that's the stable structure, and all bases are equally separated, and a inhomogeneous stretching where we have a cluster of 2, 3, 4, 5 bases stacked and then a big gap. It's out of the question that you have, let's say, more than 5 bases stacked, because then the gap we need to reach the 50% extension is it will make the backbone snap. So just by simple force consideration, you could tell something about limits here. But your question is very good, which extends to how nature uses basic physics that we have around us, and I think that has to be discovered by man.
We have a question
Okay.
from the internet. Is that Xavier?
So the first one is a general question. What chemicals are challenging systems with flushable designs, and has COVID cleaning also thrown off our water research? That's from Betsy online.
The other question is about antibiotics in the water. Antibiotics and pharmaceuticals.
I guess I could take a first stab at that. And I'm curious from the others about how COVID has affected their research programs. So the pandemic is something that water chemists have been anticipating for a while. Probably 15, 20 years ago, there were efforts to understand what would happen if there was widespread use of a drug like Tamiflu. And at that time, people realized that the levels of that pharmaceutical in wastewater would be so high that they might affect the fish downstream of wastewater treatment plants. But we didn't use Tamiflu for COVID because we didn't have a drug at that time, and so none of the drugs have made a problem. Antibiotics are a concern in wastewater, but actually the real concern related to antibiotic resistance is the antibiotic resistance genes. And so some of my colleagues up here probably understand that better than me. When lots of people are taking antibiotics, there are antibiotic-resistant bacteria around, and they undergo gene transfer. And the concern is that those genes will be transferred to pathogenic microbes. And then finally, like everyone else's lab, our lab was shut down during COVID, and in our case, it gave us a chance to kind of reassess where we were going and have some discussions about new topics. But many of my colleagues have started to become COVID detectives, and so people who have these wonderful microbiological tools have developed these ways of sampling sewage to understand when outbreaks occur. And if you've ever looked online and heard about people talking about whether there was a COVID outbreak, let's look at the sewage, not the hospital admissions. That's an outcome of kind of us being shut down for COVID and wanting to make a difference. It's that people who have these wonderful PCR technologies have found that the sewer system is an integrated sampling of a community, and you can find out whether a disease is moving in the community. You can also find out if people are taking illicit drugs. You can find out if people are stressed because you can measure stress hormones in the sewage. So what's being recognized now is that we can be surveilled through our sewage system. We can understand public health risks through the sewage system, and we can also understand levels of stress and happiness through the sewer system. So I think for many of my colleagues, not me, it opened up a new research world. I'm kind of curious with the others about their thoughts about
Andrew, did you want to comment on that?
Yeah, I mean, it's been a really great opportunity in a way. It's an odd thing to say, to use some of the technologies that had been sort of on the way to being developed to look at the sewage and the outflow. I do think if we know what the pathogen we're looking for is, there's some very sensitive assays, and those are quite valuable. There's another whole component to it which is going to need a lot of work, and people who are high school students now may be doing this when they get to be scientists, is how do we interpret things that we don't know that we find? We need to be able to find pathogens that we don't expect, that we don't know in the sewer system, and anticipate that there might be a problem. And that kind of science is something that is really in its infancy, but would really be valuable. So I will leave that as a challenge to all the high school students out there to be able to interpret the data, which comes in very voluminously in terms of what that means in biology and human terms.
Andy, I think it would be interesting to hear what you think about the hepatitis delta. You said normally it's rather harmless, but in combination with the hepatitis B, this is something we don't understand.
Right. So this is a system where there is a relatively harmless RNA molecule that is sort of a viroid type thing called hepatitis delta. In the presence of another virus infection, hepatitis B, it becomes quite nasty and pathogenic. We don't totally understand that, and then the consequences of that are that there are many interactions in biology and virology that we don't understand well enough that we have to be aware of. And so it's another area where monitoring the environment is going to be important to know what's there in order to sensitize us to things and what's there that we might react to more severely. So.
We'll go to Abdallah. You have a question?
My name is Abdallah. I'm an undergrad at Professor Yaghi's group. I have two questions. The first to David and the second to Professor Andrew. The first one, how feasible is electrochemical oxidation for wastewater treatment? This is to David, if you have any idea about that.
Yeah, so the question of using electrochemistry for wastewater treatment is intriguing, but I showed you those huge volumes of water that we have to treat every day, so it becomes a mass transfer problem. And so the limitation right now on electrochemical water treatment is that you have to overcome the surface to volume ratio challenge, which is something easy to do in the lab, hard to do in the field. And so especially when people still use flat plate electrodes or just like something that looks like a pH electrode. So as we move to three-dimensional electrodes, we can start to conquer that surface to volume ratio challenge and envision a point where we have electrochemical water treatment systems that are manufactured in modules the way reverse osmosis cartridges are today. I still think we're a decade away from that. That's why we're focusing on these small-scale applications where you're treating only the amount of water you need to drink, or you use it for industrial treatment of small volumes. I think that's constantly the challenge.
Okay. My question to Andrew is, are there any ethical considerations you have in mind when working with viruses, proteins, and so on? Ethics, does ethics come here in any place?
So the question again? Sorry, I missed it.
Are there any ethical considerations when working with proteins, viruses, and so on?
There are many technical considerations in working with them. I mean, one thing is that most of the molecules are very unstable if they're not in their normal environment, and so they'll fall apart. They'll get degraded. And so you have to be very careful to retain certain things, especially proteins, because they get denatured. The other half of that, which is almost the opposite thing, is you do have to be worried about safety too. And if you're working with materials like sewage, there could be dangerous things there. And so we in some cases will decontaminate them with some of the same compounds that were being talked about here before we actually start to work with them in a serious way, because we don't want to get infected by them. And once they're decontaminated, their infectivity is going to drop dramatically. So.
How about a question for Professor Brewer? Could you describe what your work is like? Because it's so different from most of the scientists we're hearing from today in a lab, that you're out in a ship, and maybe for some of the students here, they'd like to hear about an alternative scientific life. If you could just describe a little bit what it's like.
Oh, gee. Well, I gravitated towards ocean science because I craved adventure, and the chemistry courses I was taking were just dull. And I found an opportunity to do something different, and I've never regretted it. It takes a lot of imagination because if you're not careful, you can finish up doing very ordinary things. So we've been good about creating new tools, finding new ways. I'm kind of proud of the lab that we all built. Modern DNA instrumentation, it can be left on the seafloor, put on a buoy, survey the ocean material as it goes by, environmental DNA. We can use lasers to probe things never thought possible. It's an adventure. It can also be hard. You can be humiliated by an O-ring when you put an instrument in the ocean and the ocean gets in the instrument instead of putting the instrument in the ocean. It's not the easiest way to do science, but it's very rewarding. If I look at the future, we were the first to write papers on what is now called ocean acidification, and I took hell for writing about that. I'm not kind of concerned about the loss of oxygen in the ocean. We will have a lower oxygen ocean. In the North Atlantic, there's no problem. There's plenty of oxygen. But off the west coast of the United States, it's going to be messy. The oxygen concentration right off my lab at about 600-700 meters depth is less than 10 micromolar. We will go anoxic a few hundred meters down off California. The IPCC doesn't do very well with that. In general, the IPCC reports are ultra-conservative. People write scare stories, but the reports themselves are conservative. And I think we need to take a fresh look at some of the problems coming up.
Thank you. Well, there's still so little known about the ocean. If you compare it to what we know about the land, et cetera, why do you think it's taken us so long to focus our attentions in that direction when it's such a big part of our planet?
Well, it's difficult to do. It's a long way away. My wife can tell you, it means long hours away from home and things. It's difficult. You have to pick your problems carefully. You can't say, I want to go to see and measure something. You have to know where to go. You have to know what you want to measure. You have to phrase your questions precisely. And that's been hard. We don't understand enough about what I would call molecular fluid dynamics. Richard Zare understands molecular dynamics. People who study the physics of fluid flow, fluid dynamics, water flow on a large scale. The space scale between, say, 1 millimeter and 1 nanometer is the twilight zone. That's where organisms and water meet. And we need to know a lot more about how they do that. I would also, thinking of Bengt's talk about many biological systems are hydrophobic. I once, just for fun, and our intern, Kennedy, was with me, we tried to measure how much water there was inside a jellyfish. They look beautifully transparent and they flow. We did a crazy experiment. They contain no liquid water. The transparency is all due to their molecular material. They are dry inside. What we did was, Kennedy can vouch for it. We went out to sea, we caught a large jellyfish in a trap, and then we caught it in there, we closed the lid on the container, and then put in some argon gas and pushed the water out. And the jellyfish settled onto a metal plate, an aluminum plate full of holes. We then had a flattened jellyfish, like these flat Stanley cartoons. It just lay there, the poor animal. And then we put the laser through the glass and tried to observe any water signal inside the carcass of this poor animal, which was now drained of water and dry. Got nothing. We then put the water back in, and the animal didn't really swim away happily, but it didn't die. One of the nice things is, I showed one slide, I think, I was pointing the laser at a little fish. That happened when I took a bathroom break from the lab, and the student said, let's look over here while Peter's gone, there's a little fish. And I said, guys, get back to work. Let's become a very popular image because it shows no fish were harmed in this experiment. we found at great expense that the red color on the scales of rockfish is made of beta-carotene. And I had to say, guys, cut it out, you're just having fun. So, but yeah, you can find things out. I think Bengt is correct. That is how we understand how life survives in a dense aqueous medium like this, particularly at high pressure, is a puzzle.
Peter, I had a question for you on your high pressure experiments. You know, ice floats because it's less dense than water, and
Yeah.
the reason for that is that the hydrogen bonds space the oxygens away from each other, and under pressure, that network of water or ice, if you want to call it, interpenetrates or gets another framework to interweave to fill those spaces.
Yeah,
So that now you have double the density and doubly interpenetrated structure sinks. And do you see any of this on the? I mean, I guess this is also a question for Sekely. Do you see the molecular analogs of these where you have in your hexamers and so on? Do you see any kind of two hexagons woven or partly woven as a way to fill space? Because that affects your interpretation of the pressure results.
Yeah. Intuitively, we know that's there. That is, as we approach the freezing point, the water starts to prefer forming hexamers, which are lighter and so on. We can't see that with the really crude, simple Raman techniques we take to see. Richard Sekely can with his fancy systems. What surprises me is, first of all, we treat pressure rather badly. It turns out to be a log function, not an exponential, but it's not a linear function of depth. The equations for describing how pressure works in the ocean are really bad. We need to do better on that. I do think we're correct. That is, Le Chatelier's principle works. As we go deeper in the ocean, the less compressible species start to interfere with the equilibrium, which Sekely is correct. They're the planar forms. They help form methane hydrates. They will have consequences for marine life too. But you know, I found myself to be a one-man band here. I wrote a paper recently trying to talk about the gap between fluid dynamics and molecules. And it was roundly rejected. Basically, we said, we don't want your stinking molecules here. This is crazy. We don't like it. And what I detected was almost a fear that by introducing this topic, we were undermining religion or beliefs. It worries people. So your questions are very good, but you're into a black area between earth sciences and chemical sciences, which it's a battleground.
Okay, well, since the high school students are hesitant to ask, I'll ask a question.
Well, Shuguang had a question.
Oh,
Maybe he
all
could
right.
go first.
All right, you go first, okay.
Well, one scientific question for Bengt, and then several questions like 5 years old for any of you. The question for Bengt is, this benzoic acid, if stuck in water, then form the ring stacking just like DNA. You may be able to take FM, take this sample out after settlement or centrifugation. You can see the growth, the chain growth, the diameter, this two ring could be the same as the two molecule. You can be measured experimentally. Okay, that sounds quite hopeful you can do it.
A brilliant suggestion, but it doesn't work. You know, the backbone of nucleic acids, one reason we have the backbone is to keep the order of the bases. That's the information content, that's the basis of the genetic information. But there is another reason, and that is that the backbone serves the purpose of entropically making the concentration high. And this is why the stacking occurs. So if you take benzoic acid, you will, when you increase the concentration, you have to lower the pH, by the way, otherwise you have the benzoate, which dissolves. The crystal you form do not show stacked benzoic acid. And we have done this with adenine as well, and you would expect that the adenine would stack. They don't. They form long chains which are not stacked. So removing the backbone removes something very important in this creator of hydrophobic environment.
Well, your hydrophobic catalysis could come from the stacking different molecules.
That's right.
Yeah. Okay, now 5-year-old question. And a human need to drink very clear and treated water. But animal, dog, just drinking water on the ground. An alligator drinks water so dirty. We in Africa, we saw the elephant, this animal drink water so dirty, it's unbelievable. Human can never drink that. Why they don't get sick? Ask for all of you.
I mean, there's a couple things there. I haven't asked the dog or the alligator if they get sick once in a while, and they may have a higher tolerance for it. But I think also there's the fact that the immune systems are different in different animals, and the relatively long longevity of humans may be balanced with a little bit less effective immune system in some cases, and so we may be relying on our water being clean and human capabilities to find clean water more than a dog or a hippopotamus would.
Well, they're not drinking tap water, that's why
I'll just
they're
add that
okay.
my dogs had Giardia 4 or 5 times, so anyone who lives on the interface between people and deer, don't let your dog drink out of the gutter.
Okay, Peter?
I'd like to ask a question of the audience. We've heard several times during this meeting stories of artificial intelligence, of mining databases, of getting artificial intelligence to give us answers. I give a talk on the efficiency of microbial swimming. The literature's full of, they only work at 1% efficiency. What would an artificial intelligence system tell me if I was to ask it, how well do microbes swim? Do they think of a good answer like we tried to do, or do they repeat what's in the literature? And this goes back to, I think, a quote from Picasso about computers. He said they're useless, they can only give answers. Who asks the questions?
Bengt, can you answer that question?
No, I'm still thinking of the dog and the alligator. I think that we might be barking up the wrong tree. When we say that the water is polluted, what do we mean? I mean, it looks polluted, but it might be good for the dog. I think it's more scary when we work on refining machinery that conserves the water, so it's looping around. Then we have sort of a living system where we sort of could, we have the problem of the bacterial getting resistance and so on. And also including all the antibiotics that will circulate. I doubt that this kind of water, which I may call grey water, would be so good for the dog. But when the dog goes to a water hole like an alligator does in the water might look bad, but I think the dog is critical. He will not drink toxic water.
I mean, I might push back a little bit to say that the perception that water that's used over and over in a building or in a city is cleaner than water coming out of your tap from another source, like a reservoir, is incorrect, because there's no body of water on Earth that's purely pristine. And so when we look at many of our surface water supplies, they're dirtier than the treated sewage, and the treated sewage is going through reverse osmosis and UV peroxide afterwards. So it's kind of like one of the things that's happened to me in conducting research on water recycling has made me think about how much confidence you have as a chemist in water quality versus this long-standing cultural norms against interacting with your own waste. So civilization teaches us not to interact with our waste, and that's mainly because of pathogenic disease. But when we develop the technologies to purify water, whether it's on the International Space Station or whether it's in a city with advanced water treatment, we have to question ourselves whether it comes from, our reluctance to do this comes from longstanding cultural taboos or whether it comes from real science. So in the case of antibiotics, part per trillion levels of antibiotics may not be a concern with respect to selective pressure, and likewise when people talk about pharmaceuticals or hormones at 1/1,000,000th of a therapeutic dose, where if you touch a table, you might acquire the same dose from someone who had it on their hand when they took the drug. It gets to be a place that challenges us as scientists to say, at what point do you have confidence in the technology, and how do you slowly step the technology through and use it? And so we've worked with social scientists who think about public perception of technology, and this is one of those areas where it's possible to go round and round in circles, and I'm not even fully convinced yet. I'd rather drink pristine snowmelt water, I give you that, but I'm not convinced I understand all of the science versus the cultural taboos.
I'd like to ask some questions about water purification. I've been working on solar-driven water splitting for many years, developing catalysts to oxidize water to oxygen and reduce water to hydrogen. And I've looked at a lot of different systems, maybe metal oxides with semiconductors with high band gaps. Titanium dioxide, for example, radiated with UV light, does make hydroxyl radicals. And I wonder where that stands now in water purification. I take it it's not the TiO2, which is cheap as dirt because it's white paint. I take it that's not economical now in the water purification game.
So if you want to have a high impact factor and a highly cited paper, you can take some TiO2 and put in a beaker, and you'll write a paper, and other people studying TiO2 in a beaker will cite your work. But 40 years after the first discoveries of using photocatalysis with titanium dioxide to break down organics, we've yet to have a practical water treatment plant. And part of that is this huge engineering challenge of the large volumes of water that have to be treated, the separation of the titanium dioxide from the water, the fouling of the titanium dioxide. So we wrote a paper a couple of years ago.
I figured that must be the case.
Yeah,
it's not used.
There's a paper that we wrote in Environmental Science and Technology. It was called Sunrise or Sunset, the Future of Heterogeneous Photocatalysis. And basically, new technologies undergo a period of hype, and then a period of disillusionment, and then a period of adjustment where people decide what they are. So titanium dioxide photocatalysis is finding applications outside of water. People put it on coatings on windows to break down the dirt, or in air purification systems so that volatile chemicals in the air break down with sunlight. And the water areas turned out to be a much smaller niche that can't compete with ozone and UV peroxide because of it happens in the bulk water and not at the interface. And so I stayed away from electrochemistry for many years because I felt like that problem of the mass transfer and the interface would always be the death of any heterogeneous process. I'm getting a little bit more optimistic about it as people develop three-dimensional systems, but I'm tiptoeing in with these small-scale systems for just that reason.
Would it be any advantage to moving from UV light to blue light?
No, it hasn't been. I mean, initially the UV problem was one that you needed mercury lamps, but now we can make LEDs that can work in those ranges, and so that's not it.
You can do UV with no problem. One final thing is that in 10 years or 15 years from now, when we have these massive electrolyzers driven by solar arrays that will operate in hundreds of amps to make enormous amounts of hydrogen and oxygen from sunlight and run them back through systems to make electricity and water, will that water be useful at all, or is that just not economical?
Yeah, it's too small a volume still, so.
Too small a volume?
Yeah, so we've worked a little with EPRI, the Electric Power Research Institute, and I think
Okay.
the challenge is getting water that's pure enough, because as you know probably, you can either do it with ultra-pure water or seawater, and that challenge is one that I think is coming, the need to make this highly purified water to run those electrolyzers.
Thank you for those comments. I thought your lecture was terrific.
Thank you. Thank you
Harry,
so much. We're going to.
I have a question for you.
Okay.
You know, to produce green hydrogen, you use electrolyzers.
Yeah.
But the efficiency is still, for reasons you know, quite low.
Yeah, well,
what kind
not
of
so
catalyst?
low, but medium.
Yeah, 50%.
Not so bad.
No, but how could we increase it? What catalyst would you use?
Well, now the whole deal in the last 10 years has been trying to replace iridium oxide as the water oxidation catalyst and platinum as the proton reduction catalyst. And these have now been replaced, are being replaced by, on the cathode, with nickel phosphide or cobalt phosphides, which nanocatalysts, which now are just as efficient as platinum for hydrogen production. And on the anode side, there are now manganese, antimony oxide catalysts that are as good as iridium oxide. So the whole thing has been to replace these extremely expensive rare elements with earth-abundant elements, and that's now happening. And they're quite efficient, and so I think we're going to see a lot of big electrolyzers. They're being redesigned now because the old electrolyzers are very inefficient. But they're going to be much more efficient, and they're going to be driven by big solar arrays, and it's going to be, I think, a new world, but I understand why the water that's coming out is probably not going to be that helpful in the water purification business.
Let's, thank you so much, and let's take a question over here. Go ahead, Zach.
Oh, hello. So I'm Zach, and I'm a student from Professor Yaghi's lab. So I have a question for Professor Sedlak. So my question is, during your talk, you briefly mentioned the concept of net zero water homes, which is very intriguing and fascinating. So my question is, you comment on that, like this idea is feasible, like on most of the North America and Europe. So I wonder, like how you calculate, the amount of rain that like a certain house in a certain place can get, because I'm thinking about place, let's say here in Berkeley, we get some precipitation in February and March, while when we move to August, so the raining days become much less. So like how could you comment on that?
Yeah, thank you very much for that question. The paper at the end, Bezbera et al. That came out in 2022, we chose 6 cities around the world, and we chose them with a range of different climates. And you're right, the climate that's most challenging is the west coast of North America and west coast of South America, where we have these extreme Mediterranean climates. And our calculation was that a rainwater system in those environments would only provide water for about 6 months per year for a tall building. For some small household systems, you might make it through the whole year. And so one of the things we're talking about now is whether you could use some of the gray water from the house and take that up to drinking water quality. The other thing that we talk about is, this aversion to bottled water and trucked water. In much of the world, people rely upon tankers to deliver water when they don't have it. And finally, when I'm feeling really like on a sci-fi day, I start talking about Dune and tell people that every day when I bring my 50 kilos of groceries for the week up to the house, it's not dehydrated food, it has water in it. And if you thought about the fact that when you bring food into your home, it comes with water, if you could make that water cycle tight enough, you're probably getting 4 or 5 liters a day from the food you eat. But that's way speculative. And I think this afternoon we can talk about whether atmospheric water harvesting is another path as well.
Thank you.
We have quite a few questions here, so go ahead.
I had a question about.
I recommend quick questions and try to have quick answers to get through some of them.
Of course. I was going to ask what the next step in water recycling is, if the problem lies more with like research, finding out more research about it, or more like the economic drawbacks of this new system.
So the centralized water treatment systems are here, and they're going to happen. So Los Angeles, San Diego, Orange County. Orange County already recycles all of its wastewater. Within 25 years, Los Angeles and San Diego will too. The Bay Area's proposed a recycling plant at Palo Alto. So this horse has left the stable and is going to happen. I think the two places where there are largest challenges are in middle-income countries where it's still too expensive and we have to keep dropping the cost of the technology, and in rural communities. And that's why we're starting to look at household scale and net zero water homes, because it's still impractical to do it outside of the scale of a city.
I forgot to mention, I meant to say in terms of in-home water recycling systems.
Oh, it's reliability. I mean, you know, if it fails 1 in 1,000 times, that's a public health disaster. So I think sensors and actuators and artificial intelligence, all these things will hopefully someday make these systems as reliable as the gas heaters that are in our homes and the electricity systems that are in our homes that are dangerous but don't hurt us. Right now, there's still too high a chance of failure. We have to iron out all the bugs.
Would this also impact positively, like areas with, I guess, like water compromised areas, like for example, reservations, low-income areas?
Yeah.
Would they be positively affected with like these in-home systems that would ensure?
Yeah, that's what
better
we're
water
targeting.
quality?
Places like the Central Valley, like Allensworth, which is like the poster child for this stuff, or the colonias, or the reservations, where these water issues won't be solved with centralized water systems. So they could be part of it, but you can't have an operator present. It has to be autonomous.
Okay, thank you very much.
Okay, go ahead.
With the high amounts of synthetic nitrogen-based fertilizers being used in large-scale agriculture, are you finding an increase in nitrate contamination of water? And is reverse osmosis a viable option for removal, just in terms of that?
Nitrate is the most common drinking water pollutant in California, mainly in the Central Valley. It's that way throughout much of the United States, some of the agricultural pesticides too. It's not changing so much. Farmers are getting more efficient. Precision agriculture allows us to apply nitrogen fertilizers in a way that minimizes pollution, but it's still a pernicious problem. And so reverse osmosis is not the first choice. Usually it's some sort of ion exchange process that people go to because you don't want to create this concentrate. And you don't want to change the chemistry of the water. So there are opportunities there, and I think there are tremendous opportunities in biological treatment to get nitrate out of water.
Thank you. Next, please.
Hi, my name is Kelsey. I'm a third-year undergraduate here at Berkeley, and I used to work in membrane research in Martin Anderson's group at Chalmers, actually. And my question is also for Professor Sedlak. Sorry to play favorites, I guess, but my question surrounds kind of more the process of reverse osmosis, because my understanding of the process is that it's really like highly energy consuming, and so I was wondering if your group has done anything to kind of address that or investigate other methods.
So a modern seawater desalination plant uses between about 3 kilowatt hours and 4 kilowatt hours per cubic meter to move water. Pumping water up and over the Tehachapi's into Los Angeles uses about the same. So we've reached a point where the energy that we're using to pump water around is comparable to what modern seawater desalination plants use. And so the push now is to, when someone builds a seawater desalination plant, to couple it with a wind farm or a solar farm, because it's kind of a known amount of additional electricity use in the grid. And so like in Perth, Australia, they did green seawater desalination by building wind farms and solar farms. I think similar things will happen. California's grid, which is going to be mostly greenhouse gas, low greenhouse gas in the coming years, is a case study in that.
Great, thank you so much, and thank you all for your presentations.
Thank you. Next question, and this will be our last question, and then perhaps you can just have a few comments.
Okay, this is for Professor Sedlak as well. So sorry, this is not super scientific, but you mentioned that the Salesforce Tower has its own like water treatment, and you said that in a couple years you'd feel comfortable drinking out of it. So like, when do you think as a society we'll change our perspective on drinking treated water?
When I started working in this area of potable water recycling, there were a few projects in Southern California, most notoriously in San Diego, that died because someone stood up at a public meeting and said, I'm not going to drink out of my toilet, this is toilet to tap and all these things. And then the 2015 drought happened, and then another drought happened. And now when you go to public meetings in Southern California, people are saying, sign me up. And so we've studied this process with some social scientists from the ETH in Zurich. And as I mentioned before, it's a process called legitimization of technologies that happens when the public decides whether something that is unfamiliar to them is something that they should embrace. We've seen examples when technologies have successfully undergone legitimization, like air travel, for example. Scary stuff. People died in the early days, but we all love traveling by air. We've seen nuclear power rejected by most countries on Earth because of concerns about safety. So potable water recycling is undergoing this process of legitimization now, and judging by what's happened in Southern California, it's crossed that threshold, and we're going to see billions of dollars of investments in the coming years. The only thing I think that could derail it is like a high-profile misstep, which might change the public's opinion. But this is kind of a great place for chemists to think about this idea of just because you make a technology doesn't mean society accepts it, and that's where we have to learn to work with social scientists and psychologists and political scientists to understand whether the public accepts the technological innovations that we've come up with.
Okay, thank you so much.
Do we have any more time?
A couple of minutes if you'd like to.
A couple of minutes, okay. I have a question for you, Andy. Could you comment or speculate on the generation of these circular RNA? What is their role? Is it understood what their biological function is?
Right. It's a good question, and the answer is we don't know, of course. So the speculation would be that they would potentially be either components of the cell which allow information to be passed from cell generation to benefit the cell, or that they would be infectious agents. And there are examples on the DNA level, which is probably the best analogy we have of plasmids, that are both things. There are circular DNAs that are components of the cell machinery and that probably contribute to what the cell does, not that we always like that as other cells. And then there are ones that are viruses that can interfere with the cell. And it's all a question of whether they're infectious, and I think the question is what those proteins do. Do they mediate infection, or do they protect the RNA and allow it to replicate? And so that's a question that we and I think others probably would like to get at.
Thank you. I have one last question, and I'm asking it on behalf of the high school students, because whenever I give a general talk, somebody stands up and says, how do I discover something? And I won't tell you my answer because I want you to come up with your own. But can Andy, maybe since you have the mic, maybe you can tell us how you discover something.
There's two questions. How did people discover things, and how can you discover something new?
No, how do I, as an unprepared researcher, go in a lab and discover something? Right? I'm a high school student. I'm interested in finding out more about things, and I want to discover something. I want to be as famous as you are.
There are a lot of questions to that, and I think one of the answers is you ask questions, and you interact with colleagues and friends and other students and faculty, mostly the other students' part, and you see what's interesting to them, and you communicate on things, and you work together to ask questions that might give unique answers. There are certainly many, many questions to answer, and often the issue is we can't answer them. And so there's plenty to be done and plenty of need also for people to be asking questions and discovering things. So go and do it, and you'll enjoy various parts of it, and sometimes you'll discover something very small, like there's one other molecule here that no one noticed, and sometimes you might discover something bigger, and you have to get pleasure out of both of those.
So get it done,
listening,
doing,
talking,
experimenting.
Interacting, yeah. fun.
Bengt?
Well, I see another dimension of it, and that's that you begin to think when you observe something unexpected. Normally when people see that the result in the test tube and didn't go as well, they throw away the result. I think you discover things when you really observe something that is counterintuitive or seems wrong. And I think historically that fits relatively well. I think when you discovered the RNA hybridization, somebody else might have not have thought about it.
That's true. Measurement is also very important, and understanding measurement and measuring things carefully and knowing what you can and can't measure. Sure. And maybe making better tools to measure. Those are all very important.
Peter, not the definition of discovery, but how do you discover? How an unprepared researcher discovers?
I'm not sure I know. I get an awful lot of tenure files to review or promotion files, and universally I look for the characteristics of growth and development. If you see somebody who's doing the same thing now that they were doing 40 years ago, I don't like it. So you want people to show the ability to develop within a field and then grow beyond that because they find something new to look at. And I found that time and again, you milk a problem, you've gone as far as you can, and you think, well, what's next? And you look around and you find the next things, and it usually follows logically. So it's the prepared mind that's allowed to wander a little. Don't get stuck on one thing. Feel the need to grow, and you'll get it.
Thank you.
Thank you.
Dave?
Favors the unprepared mind, too. So if you don't know it, still do it.
I guess for a high school student, I think it's a little like, think of yourself as learning a musical instrument or learning how to speak a foreign language. The first part of getting through the Suzuki violin book is kind of monotonous and boring, but at some point you develop a certain mastery and proficiency. And time and time again, we see the people come up with the most creative ideas are the people who've just mastered something and ask that question, why? And when people get set in their ways by years and years of seeing the same thing over and over again, they're a little less open to the why does that work? And so I think the hardest part about being a high school student in chemistry is probably you're still learning your syntax and your vocabulary and your grammar, but you're rapidly approaching the point where you're going to have the most productive and creative years, and it comes from asking why and questioning when people try to tell you things that don't make a lot of sense.
Thank you very much. Okay, Lorie.
Thank you all. We really appreciate this morning's session, which has been excellent. So